Thank you, Pasi. So now we'll hear from Don Harvey about some of the drawbacks to rapid drug development.
I want to thank the organizers for inviting me today, Dr. Topalian, Dr. Shilsky, and Dr. Pence from Emory, who recommended me for this panel. I also want to thank the speakers who came before me because each of them has, I believe, nice examples of some of the challenges that have come forward with drug development in early phase trials. For example, pembrolizumab, the question of 2 versus 10, it's a 5-fold dose change, which certainly has its challenges. Vemurafenib and its predecessor had issues with formulation, which I think are becoming more prevalent as we look at some of the rapid accruals. And then finally, the examples of ruxolitinib in terms of its active metabolites that had off-target effects on IGF-1R and QT prolongation. So my job today is not to rain on any parades, because I do believe that there's a lot of value in accelerating the drug development world that we live in, but that we do have some challenges ahead of us. And those two big therapeutic directions have already been mentioned here today, and that's where they're going down a path of targeted molecular therapies, and those can both be small molecules as well as monoclonal antibodies, but also immunotherapeutics. And we've seen challenges in combining those two classes already. When you look at the combination of vemurafenib and ipilimumab, that was the first phase 1 trial I can recall in New England Journal of Medicine that was a negative trial. It was stopped early because of hepatotoxicity. And so there were overlapping toxicities there that weren't necessarily predicted as that trial got open. And then finally, the idea of seamless drug development, which is talked over and over, which I won't go into too much here. But there were certainly early examples of this seamless approach, which have led to new molecules getting to the market for patients, which is what we want. And the pembrolizumab activity has already been seen and mentioned here today. And certainly in Atlanta and Jimmy Carter, we thank pembrolizumab's activity because there are a lot of Habitat for Humanity homes that have been built after treatment. And in the end, we have a much more efficient development and much more lower sample size needed to define activity of drugs across different areas. There's a nice paper by Dr. Pazdur and his group from FDA that talk about seamless drug development, some of the summary of questions within the seamless world. And as has already been mentioned as well, seamlessness is not going to be applicable to every single molecule across the board. And as was mentioned by Dr. Rubin already, even at Merck, they're not necessarily taking a seamless approach with all of their new compounds because there are questions around that. And so I'd encourage everyone to read that trial. There are gaps, and so we need to mine those gaps in seamless drug development. We have a decreased trial population, and so therefore the N that's coming of patients onto trials, and therefore in the post-marketing world is lower potentially than it was in the previous paradigm of phase 1, 2, and 3. We have certainly a rapid acceleration of dose derivation, and the issue here is that you've got the fountainhead of a dose that's created in the phase 1 setting, and that fountainhead dictates everything subsequently. And if that fountainhead is wrong, the amplification of error is certainly increased across the board, but there's also pressure to move that dose forward and adopt it for licensing trials. And finally, I really want to shine a light on some of the clinical pharmacology studies that are gapped within this seamless approach. I went back and looked on the FDA website and sort of summarized a lot of the oral agents that have been FDA approved over time. And from 2006 to 2010, we had this range, and then after 2011 and beyond, we had a banner year in 2015 for oral agents that were new molecular entities that were developed. And so these are all incredibly important advances in their own rights and in their own ways, but it creates also a situation where we have lots of opportunity for potentially getting it wrong, and I'll argue that we have gotten it wrong in some instances. And one of the biggest issues is dose determination, which has also been talked about, and that's that in the phase 1 and perhaps in phase 2, although rarely, that's really the fundamental process of defining a dose for a compound moving forward for subsequent trials. There's registration-directed doses, and if you really fundamentally philosophically think about it, we're trying to understand pharmacokinetics and pharmacodynamics in early phase trials where we're escalating doses in small numbers of patients. The reality is that some of the best ways to understand patient covariates and variability in dose and exposure are when you have a fixed dose in a fixed population, and that's not how we routinely develop drugs in terms of understanding the correlative clinical pharmacokinetics and pharmacodynamics. And then finally, in the post-marketing commercial access, I think one of the biggest challenges we have is in this space here, and I'm going to go through as well some of the post-marketing commitments that have been expected of companies and compounds to be done. I can tell you that running a phase 1 unit and program at Emory, it was a lot easier to get patients on an osimertinib CYP3A4 clinical trial before its FDA approval than after its FDA approval. Similarly with food effects study, if I can get a drug, if I'm a patient and I can get a drug prescribed to me by my oncologist in the commercial setting, or I can go on to a trial where I get to eat a Georgia-style breakfast of 3,000 calories to understand fat effects on absorption, I'm probably going to just get the prescription. So the accrual becomes much more challenging over time. Whereas with osimertinib, we ran a lot of the ClinPharm studies at Emory, and we had patients lining up over and over before the drug was approved. And so the accrual does become a challenge pre and post-marketing. So in processing of dose finding, we have preclinical tox data that leads to the first level of dose within any group. And one point that's been brought up already today, and I think one that's growing an issue, is formulation. When we ask patients to not take any antacid at all forever while you're on this drug, and oh, by the way, the drug causes nausea, it becomes a bit challenging. And patients have to space multiple hours. I think bioavailability has taken a hit over time in our formulations as well. We have opportunities for improved formulations when we have 5 and 10% bioavailability, a 12-pill burden for a given patient on a given regimen, which you can probably think of in a given day because of poor solubility. These are drugs that are certainly improving the care of patients, but do have clinical implications. Dose derivation is certainly more challenging with oral than with IV therapies. As a pharmacologist, I like IV therapies. They're 100% bioavailable all the time, but it does become much more tied to a clinic environment. We've talked about maximum tolerated dose. I want to thank Mark for bookending this in terms of MTD and its role as well. But in general, what we do in trials is to find dose for a population. But the challenge is that in the clinic post-approval, dose is not for a population, dose is for an individual. And so those population doses that are defined are fine, but individual dose adjustment is empiric. And so hopefully we'll come to some thoughts on what we might be able to do to improve this paradigm. A big challenge is the dose exposure and effect relationship is rarely known a priori before you head into a trial, a first-in-human study. We don't know what dose leads to what exposure, leads to what effect, positive or negative, in terms of area under the curve or other PK measures of exposure. We have had a very high rate of dose reductions in subsequent trials, and I'll go through some of that data where upwards of 80–85% of people have had dose reductions required in phase 3 studies of fixed dose agents. We also fail to identify patients who may benefit from lower and higher doses. When patients progress on a given therapy, particularly if it's an oral therapy, we tend to assume that it's because the cancer got worse or another issue came up in terms of resistance. The reality is that they may not have enough high enough exposure for them to develop activity, and perhaps a role of therapeutic drug monitoring should be returned to in those patients and in those drugs where there are extremely variable exposures after a fixed dose. And then finally, for targeted agents, the optimal biologic dose certainly may not be the MTD, and dosing to where we make patients sort of have excess toxicity over time may not be the right approach either. Chronic therapy-limiting AEs may not be incorporated in dose decisions, so if patients have diarrhea that's grade 1, we don't call it a DLT in cycle 1, but I can tell you that patient who's on that drug for 10 or 11 months and comes 4 hours from South Georgia isn't excited about having grade 1 and 2 diarrhea for 12 months. The application of the MTD to subsequent combinations and populations may be questionable. We may have a more advanced population early on, and perhaps the dose that's used in a newer diagnosis group might be different and might be tolerable, and certainly in combinations. And then we rarely, if ever, explore multiple schedules in the development process of a drug. Those tend to be post-marketing questions as opposed to during and pre-development approaches. There's a nice paper published in JCO in 2011 of 77 trials at MD Anderson that looked at 450 patients. And I'll point out a couple of things about this, and that's that those of you who are in phase 1 drug development understand the process of, well, let's just get them through the first cycle, and then we'll see how they do after that. And that's really depicted here, in that if you look at dose reduction that occurs, there's very rarely a dose reduction that occurs in the first cycle, but it shoots up in the second cycle. And so patients have dose reductions in cycles 2 and 3 very commonly in early drug development. And so therefore, we should be using that data as we think about dose and its derivation subsequently, because the reality is that this is not likely a tolerable dose for the long period of time, and therefore, what is the dose that's really most expressly given over the longer cycle period? It's important for tolerability, it's important for adherence, and optimally, it's important for cancer management. So now we're going to have a little bit of a case study. I want everybody to guess these drugs, and I'm not going to tell you what they are because I think these are important examples, at least not during the presentation, important examples of places where I think we've gotten it wrong. So drug X is a multi-kinase inhibitor. The FDA-approved dose was the MTD from single-agent phase 1 trials. But in the phase 3 trial, 86% of patients required at least one dose reduction as part of their therapy on that trial, and that was either an interruption, a reduction, or discontinuation. And there were 2 dose reductions allowed. If you started here, you could either be dose reduced to 70% or to 40% of the starting dose, so less than half of what you were on originally. The PK/PD is an important concept within this. As I jokingly said during the break, you never want to be interesting in clinical pharmacology. You don't want a drug that's incredibly interesting because it generally creates more challenges. The high-fat meal increases the area under the curve by a little over 50%. It's got a half-life of a little over 2 days, and it accumulates 5-fold at 21 days. And so the accumulation at 3 weeks means that you may start to see adverse events at 4 and 5 weeks that you didn't see in the first cycle. And so you've now taken a first cycle dose and moved it and kicked the can down the road to cycle 2 with the pharmacokinetic profile of the drug. There's no relationship between exposure and progression-free survival, and that's shown here. And when you see progression-free survival on the left graph, and the exposed study months, and these are doses, so the dose you can see overlaps in terms of progression-free survival rates versus placebo in the trial. Similarly, progression-free survival and area under the curve in different quartiles was also overlapping across the board, and so dose didn't predict progression-free survival. Exposure, from a PK perspective, didn't predict progression-free survival, and so in the end, you're left with what's the right dose for this compound. The other part of it from a safety perspective is that increased exposure was decreased time to first dose reduction and an increased QT interval. And so there was a post-marketing requirement, post-approval requirement for this drug to study lower doses. If you think it's hard to get patients to go on to a food effect study, a gastric pH study, or a cytochrome P450 study, imagine trying to tell a patient coming into your clinic, well, the drug's approved at this dose, but you might get randomized to get this dose, or this dose. Do you want to sign up? So it's a difficult selling point in the post-approval world to get patients to buy into lower doses. So another multi-kinase inhibitor, 4 tablets daily, so a significant pill burden for patients. PK is listed here. There are 2 active metabolites, an M2 metabolite and an M5 metabolite, both of which are active. And so you're not dealing with one drug, you're dealing with 3 in terms of the activity there. The different formulation was used in clinical trials versus the one that actually came to the market. And so that formulation difference creates questions, at least in terms of whether or not there were opportunities for understanding different clin pharm aspects of the differing formulations. It undergoes intrahepatic cycling with 3 peaks at 4, 8, and 24 hours. The parent and metabolites are highly albumin bound. And so if you have a patient who's perhaps post-marketing in the clinic, colon cancer, gastric cancer, for example, they have an albumin of 2.7. Their free fraction of drug is going to be much higher than someone with an albumin that's normal, and therefore they may have more adverse events similarly to, or at least from protein binding and free drug. Their M2 and M5 exposure was higher in those over 65 years, and similarly a high-fat meal increases the parent area under the curve exposure by 48%. Dose interruptions were required in many due to adverse events. There were 2 post-marketing clin pharm commitments 3 post-marketing trials, and the variability for the metabolites was seen. And the FDA Clin Pharm review very adroitly pointed out that a population PK analysis hadn't been done, and therefore covariates contributing to the variability are currently unknown. And that's an area I think of improvement and possible improvement in our drug development approach, and one that FDA just released guidance on. That's looking at physiologically based pharmacokinetic models to try to get dose right earlier. And I think that's an incredible step forward to try to understand it. And so post-approval since 2001, to summarize it, we've had 28 new approvals, 11 were accelerated. Of those 28 drugs, and this is data from the FDA's new revamped website, which is really handy if you want to take a look at it, as well as clinicaltrials.gov. 78 post-marketing requirements, so that's roughly an average of 3 trials per drug. 5 of those were dose finding, meaning that the FDA-approved dose was what went on the market, but the dose finding study was all lower doses. So it's comparing the approved dose to either 1 or 2 lower doses in 5 instances. There were 30 drug-drug interaction studies, which included both cytochrome P450 enzymes as well as gastric pH effects, 19 hepatic impairment and 7 renal impairment trials, and then 2 food effect studies out of those 78. And so how can we get better at this? And the dose precision is an important point. So precision is really the idea of getting the dose right for the majority of the populations that are going to be treated with the drug. And it may be that populations that can handle different doses of drug based on their disease alone, or perhaps performance status or other clinical variables. We can certainly improve formulation. I think formulation is an area where we've got a lot of opportunity. There's a lot of work to be done to improve formulations early on so that we're not having to deal with consistent GI effects, patients with large pill burdens, low bioavailability, which may not be known until the end of a study unless real-time PK evaluation is being done. Earlier effects, and then again, the idea of physiologically based PK models I think is incredibly important if you can get all of that data and get it analyzed in silico quickly, and continue the idea of sparse PK collection in late stage trials. That's really where Clin Pharm can, I think, have additional value, and that's in the idea that everybody's getting a fixed dose. You've really likely homogenized the population more, certainly from a disease perspective, and therefore that PK can be very useful and very thoughtful if modeled appropriately. And then ask the idea about multiple doses for differing populations, which may be defined by clinical variables. And then finally, I'll summarize with this slide, and that's an idea of this same paper in 2011 of a proposed phase 2 dose model. And that's the idea that if you determine a dose in cycle 1 that is equal to the dose that's used in cycles 2, 3, 4, 5, and 6, then you're done. You have your dose that can be used in an individual patient or population for subsequent evaluations. However, if you get to a point where your dose in the acute phase setting where we define DLT is different from that in the chronic setting, perhaps a weighting approach might be used to define what the weight of that dose should be as you look forward. And if those doses are different in the acute phase versus the chronic phase, then you can look at expansions, which we've talked about, that might need to be statistically valid, and think about what they might be, or return to something which probably nobody wants to do, but would be probably the most elegant, and that's, or most definitive, and that's a randomized phase 2 study of dose. Asking the question, if you give this dose to this population, the other dose to this population, what's the right endpoint in terms of dose carrying forward, both for activity as well as adverse events. So with that, I'll stop and thank you for your time.